Imagine a microscopic war raging all around us, bacteria versus their ancient enemies, the bacteriophages. These phages are relentless, injecting their genetic code into bacteria and turning them into virus factories. The infected cell becomes a zombie, churning out hundreds of new phages until it bursts, releasing a viral army. But bacteria aren't defenseless. Over billions of years, they've evolved clever defenses. One of their most famous weapons? CRISPR, a molecular memory bank that stores pieces of viral DNA. When a familiar virus attacks, the bacterium uses this memory to guide a protein, like Cas9, to slice up the invader's genetic code. It's a single-celled immune system, precise and effective. This microscopic arms race is ongoing, shaping life on Earth. The stakes? Survival of entire bacterial colonies. And sometimes, the defense is even more dramatic than a simple cut and paste. Let's meet a new kind of bacterial hero. Enter CAT1, a CRISPR system with a radical approach. Unlike Cas9, CAT1 doesn't just cut viral DNA, it's designed for self-sacrifice. When a virus overwhelms a cell, CAT1 activates a last resort alarm, a tiny molecule called CA4. This signal means the cell is lost, but the colony can still be saved. CAT1's mission? Destroy the infected cell before the virus can escape. This is called abortive infection, a scorched earth tactic. By sacrificing itself, the bacterium becomes a dead end for the virus, protecting its neighbors. CAT1 is part of a special CRISPR team called CARF effectors, triggered only when all else fails. It's not about saving one cell, it's about saving the community. Even the simplest life forms can act for the greater good. CAT1's strategy is ruthless, but it works. Now let's see how this microscopic hero pulls the plug. Inside the doomed cell, the CA4 alarm rings out. CAT1 proteins, dormant until now, snap into action when they detect this signal. But CAT1 doesn't attack the virus directly. It targets the cell's energy supply, NAD+. NAD+ is the cell's battery, powering everything, including virus production. CAT1 shreds NAD+, draining the cell's energy in moments. The result? The cell shuts down and the virus is trapped, unable to finish replicating or escape. Hundreds of half-built viruses are left stranded. The sacrifice of one cell creates a firewall, halting the outbreak. It's a swift, total shutdown. No energy, no escape, no new infections. CAT1's self-destruct sequence is a masterstroke of microbial defense. Sometimes the best way to win is to deny the enemy any resources at all. But how does CAT1 unleash such power so quickly? The answer lies in its remarkable structure. When activated, CAT1 proteins link together, forming a deadly spiral filament. This spiral is the ultimate energy-destroying machine, rapidly depleting NAD+. The shape allows many CAT1 proteins to work together, amplifying their destructive power. It's a beautifully organized structure, efficient, fast, and irreversible. The spiral ensures the cell's self-destruction happens before the virus can escape. This discovery reveals the hidden complexity inside even the simplest cells. CAT1's spiral is a marvel of molecular engineering built for one purpose: sacrifice for survival. Evolution has crafted a microscopic weapon of astonishing sophistication. CAT1's story opens a new chapter in our understanding of bacterial defenses. It shows there's more than one way to fight a virus: precision or self-destruction. These strategies inspire new ideas for science and medicine. Imagine harnessing CAT1's power to target cancer cells or control infections in new ways. The CA4 signal and spiral structure offer fresh targets for research and drug design. Bacteria's ancient arms race could help us fight antibiotic resistance. Even the smallest organisms can teach us about survival, sacrifice, and innovation. By studying these tiny warriors, we unlock secrets that could shape the future of medicine.